Moving on to the next talk, Max Erdmann, who is currently at the University of Liverpool, but I suspect this work was done when he was in Bristol. And he's going to talk about, it's not all about the spike, and looking at mutations and replication. Thanks very much, Max.
Thanks, James. I hope everyone can hear me okay. Yes, so hi, my name's Max. Thank you for organising this brilliant event, UK-ICN, and having me. And as you can see, my talk will be about, it's not all about spike, investigating non-spike lineage-defining mutations and their impact on SARS-CoV-2 replication. So, actually, I should also say, as James has preempted, this is work that I did during my PhD in Bristol, but now I'm in Liverpool. So, if you can cast your mind back to SARS-CoV-2, it entered the population in the end of 2019, and then we observed rapid viral evolution. Variants started to emerge. First we had B1, and now we're in, I think, 20 Omicron sublineages in a very short amount of time. And we hypothesized that while there were lots of mutations observed within the spike that started to evade antibody-mediated neutralization, we also observed those in the non-spike regions of the genome. So that's ORF1ab, but also the accessory genes. And we thought, well, what do these mean? Are they simply just carrying with the spike, or are they actually adaptation to replication in humans and human cells? So to investigate this, we set out and made a panel of recombinant viruses. So on the left-hand side here, you can see those that essentially present the variant of concern spike in the lineage B1 isogenic backbone. And then we have made the panel of essentially the inverse chimeric virus, where we have the variant of concern genetic background expressing isogenic D614G spike. And we hypothesized that if they have the same spike, they enter the cells the same way, so we can normalize it in this way, but also equally the inverse is, if you have the same backbone, you should have the same intracellular replications, and differences we see are due to different entry of the different spikes. My work has mainly focused on investigating the recombinant variant of concern backbones. So initially, once we had made these viruses, we characterized their replication kinetics in our very old good friend Vero. We didn't really see any changes. Perfect, thank you. We didn't really see any changes in replication in the cell line. That was kind of not quite what I expected, and as a control I also included one where I would expect to see a difference, so the BA1 spike replicated slightly slower than the wild type. But this was kind of surprising. So then we thought, well, okay, maybe we don't see a difference in viral titers released into the supernatants, but maybe actually it's still altering cell-to-cell spread. So we then started doing some quite old-school virology and looked at the plaque morphology of these viruses. And then so that's where we started seeing some sort of initial differences. So for alpha and beta here on the left-hand side, it was quite interesting. So dependent on the backbone or the spike, the recombinants behaved either the same as the variant of concern isolate or the lineage B wild-type comparator virus. So for alpha and beta, it appeared as if their plaque phenotype was determined by the viral backbone rather than the spike protein. And then as the pandemic moved on to Delta, Delta sort of lies in between. So actually for Delta we saw it looks like the actual variant of concern backbone appears to form smaller spikes, whereas the Delta spike actually is quite fusogenic and produces larger plaques. So Delta presented as the intermediate. And then on the extreme other example, actually once Omicron BA1 emerged, we saw that this virus was almost entirely defined in its plaque phenotype by the spike protein. So that was interesting. But then next we moved on and started looking at human cell lines and their replication. These cells are derived of CACA2 cells, and I picked these particularly because they've been reported to allow persistent and chronic infection with SARS-CoV-2 in cell culture. And as it's been brilliantly introduced earlier, there's the commonly believed theory that a lot of these variants of concern may have emerged in persistent infections. So I thought maybe we would see differences here. Again, we didn't, but that was fine. We then did start to see some differences once we started playing with human lung cells, Cal-3s, that also have an intact interferon response. So here, actually, surprisingly, we found that alpha and BA1 replicated extremely well. They compare very well to lineage B1 virus, but actually beta and delta appeared to be quite severely attenuated in their replication in the cell line. And then I thought, oh, maybe this is due to the difference being the interferon response. So we then looked at the cell line again, and it is essentially the same experiment, except that in the green curves, I included an inhibitor of the JAK-STAT signaling pathway. So essentially, they behave as if the cell line wouldn't have an intact interferon response. And for the two viruses that replicated extremely well in these cell lines, so BA1 here on the left-hand side and B1, we didn't really see a difference, but actually beta was like markedly increased in its replication, so 12.6-fold compared to 2 and just about 2 for the other two viruses, and this also translated to massively increased spread in the actual cell sheets between treatment and untreatment. So it looked that actually variant of concern beta was quite severely restricted by the interferon response. Fortunately, in the lab, we had also just bought some interferon, so I thought, well, okay, maybe it's quite as easy and straightforward as simply pre-treating cells with interferon alpha and comparing, does actually then this translate to reduced replication capability? The long story short is, no, it didn't, and I was slightly surprised. So we then decided, okay, well, we don't really know where this is going. We thought it was the interferon response. Let's look a bit deeper. So we then looked at the transcriptome of Cal-3 cells infected with beta, BA1, and the recombinant wild type. And actually what then we did find was that, this is showing a PCA plot and the clustering of the samples. And we did then actually do see that the difference between these two groups, the beta mock treatment and BA1 and the wild type, the difference between those two groups was largely defined by the interferon response. So then the story started coming together, and actually what we noticed was that interferon alpha was not relevant really for SARS-CoV-2 infection in this content, and it was mainly interferon beta-1 and interferon lambda. Interestingly, when we then did some clustering analysis, the beta actually clustered away from the other virally infected groups. It clustered with the mock, and this was still true actually once the replication was recovered with JAK-STAT inhibition. So then I started to look a little bit closer at what genes actually were differently regulated between these viruses, and it really did come down to the interferon response and interferon-sensitive genes. So just before I go to the next slide, I'd like to draw your attention to this here. So beta had markedly reduced levels of RNA within these cells, and I mean, this translates to reduced infection. But what we did then notice was that even though the viral load in these cells of beta was massively reduced compared to the other viruses, it induced interferon-sensitive genes to levels that were not significantly different to the other viruses, but in fact actually for some of them even higher. So somehow the lineage-defining mutations of the variant of concern beta backbone essentially resulted in a catastrophic loss to control the interferon response. And even when we treated the cells with this JAK-STAT pathway inhibitor, for the other viruses, you do lose the association with the interferon responses and kind of immune activities, but even when you treat with this inhibitor, beta is still associated with functions such as cytokine activity, cytokine receptor binding, and CXCR, chemokine receptor binding. So then this kind of drew the whole story together, and maybe also then explains why beta never really took off like other variants like Delta or BA1. Yes, and that brings me to the end of my talk. And then just to recap what I've talked about. So It is not all about spike. The variant of concern mutations that are located outside of the variant of concern spikes, while it did not alter replication in Vero's or CACA2 cells, they did alter the spread through the cell sheet and altered cell-to-cell transmission. And particularly for beta, this then resulted in reduced attenuated replication that was partially recoverable by JAK-STAT blockade. And overall, as a virus, this resulted in a failure to antagonize the interferon response and replicate in the presence of an interferon response. So this then only leaves me to thank all the people that were involved in this group, in this work. My supervisor Andrew and David, Isabel Webb, Maya, Ed, James, and the rest of the lab. I hope I have not forgot anything. Thank you very much for your attention. Yeah.
Thanks, Max. It's a great talk. Lots of questions. Paul, you...
So the hypothesis for beta then is it was immunologically fitter because of the spike change, but held back by...
Yeah, so I think
poor
beta
interferon
essentially, where we did see kind of the localized epidemics or where it did spread, it was probably compensated by the neutralizing antibody escape that was present by mutations in the spike, because we didn't really see that much reduction in neutralization by alpha, but beta then was kind of the first one that really started to dip. But overall, the actual backbone resulted in worse replication.
So why didn't it evolve better interferon antagonism? I mean, did you have
Beta,
candidate polymorphisms that make it poor at controlling?
Yeah, so we do have a couple ones that we want to look at, and so Isabel, who'll be carrying on the project, is making individual ORF swaps. I think particularly ORF3A has an interesting mutation that results in loss of, in the loss to remodel the autophagy response of the cell. So this is residue 171, I think is quite interesting. Also, beta didn't have the NSP6 LSG deletion, so that could also be a case. So yeah, there's lots of work planned to go ahead forward, and also there's a lot more resources in this transcriptomics data set.
I think it's basically Paul's end of the question as well. So you have looked at the ORFs, but have you also looked whether you're losing kind of additional underlying genes that could potentially be expressed, like the accessory ones in ORF9b, C, etc., in your variants of concern?
No, so I haven't looked at the subgenomic RNA ratios, but I think that would be something to do going forward. So essentially, I kind of did the characterization. Now that we have picked out this virus, we're kind of digging down with the reverse genetics and investigating the individual. So what I'd like to do would be subgenomics and also like protein levels, because maybe even though it makes a subgenomics, it doesn't result in the same amount of translation.
That's great. Thanks
Right,
very much.
thanks everyone.